So without wasting time, so thank you once again for joining this introductory statistics session. So for those who don't know me, my name is Clement Chumasi. So basically, I had my PhD in mathematics at Cardiff University, and from there I went further to do my postdoc at the Oxford Statistics Department, and then proceeded to becoming a clinical trial statistician at Imperial College, and later have joined the Nuffield Department of Medicine as a medical statistician. So I'm very, very humbled to be part of the team. And then, so without wasting time, I'll just recap the outline of the presentation. So basically, we are going to recap a bit about some of the upcoming STAS teaching sessions. Of course, at the end of this particular meeting, there will be a small survey that will be sent for researchers and also for everyone to give their feedback as to what else they want in terms of some specific needs of interest, so that we can cover in some future time. And so I'll also touch a bit on some useful information about variable types and data structures and how relevant they are when it comes to trying to test even hypotheses or also trying to do statistical analysis. And I also understand that we have different levels of expertise. So in a sense, some are new to statistical analysis or statistics in general, and some are advanced users and so on. So I'm hoping to try and get an intersection of the two. So at the end of the day, I'm sure everyone will pick something new or something useful for their respective studies. And so we'll touch a bit about univariate and multivariate methods in the context of both parametric and non-parametric cases. And then most importantly, we'll talk about regression analysis, since it's often one of the most sought after kind of statistical tests for across different disciplines. And then we'll touch a bit about variable selection techniques and what are some of the statistical softwares available that one could use to perform statistical analysis, and also even sample size determinations and so on. So I just touch a bit on a few which could be useful, and then we'll introduce a bit of about the next meeting that is happening next week, and then what we expect to see. We'll touch a bit on that as well. So as I said earlier, so on Tuesday we had a first STAR session, and we had quite a number of people joining. But unfortunately, since it wasn't recorded, I'm hoping to record this one because the previous one didn't have recording right. And so today is just a repetition of the first session. So it means next week, Tuesday, we are going to now introduce the second session of the introductory statistics session. And then there will be a repetition again on the Friday. And subsequently, the goal is also now trying to get researchers to present maybe the statistics bit of their work, and to get to get opinions as to the best thing they could do, or in case maybe they have a particular hypothesis, they have a particular data, they are not too sure what can be done in order to answer some specific research questions. So the goal was to have such an instance where researchers could now brainstorm over a presenter's work. So it could be maybe 3 people presenting within a certain period, maybe within a period of 1 hour, 30 minutes, and so on. But it's still something under the developing stage. So and it's also subjected to opinions, and also we are happy to take feedbacks as well. And then there was also plans of having a drop-in session where again, if anyone has a query, a statistical query, they could book for such a session, and then we'll take it from there. But again, it's still something that we need feedback as to how we can do that to really optimize time, and also looking at the fact that we have researchers across different disciplines within the Department of Medicine. So for the introductory statistics session 1 is basically just trying to talk about like the basic or the most important things you need to know in statistics, just the basics. But of course, there will be something new you learn. And then in the session 2, we'll touch a bit on more customized statistical applications or more advanced methods that one could also effectively adopt in their respective analysis. Okay, so a question that we always know is that what statistics analysis is all about. So you realize that in every discipline we collect data, and after collecting data, we often have some specific research questions we want to answer. Sometimes the research questions are translated into research hypothesis. And so what statistics try to do is that after collecting data, we try to analyze the data, and after the analysis, the goal is to interpret the data and make inference as much as possible. But one thing we have to notice that identifying or using the right statistical method for the right job is very imperative. So on Tuesday, I gave an example that suppose you were trying to dig a small hole or dig a hole in a space which is quite confined, such that one could just decide to use a fork to dig that hole, and one could decide to use maybe a tractor to dig that hole. But imagine the consequences of the tractor, the things it could destroy at the same time. But one could decide to use a shovel to dig that hole, and it seems that at that point it's more optimal choice. This is just to say that identifying the right statistical method to do the right job is very imperative. So this is also emphasizing the need for why we need to understand the kind of data we have, the type of variables we have, and then which statistical method is very important. If we really want to now answer some specific questions, then we need to do it the right way. So that brings us to why we need to understand some of these introductory stat concepts, and that could be imperative for our respective domains. So now in statistics or in statistical analysis, often there are two things that we look at. So it means all statistical methods can be categorized into descriptive and inferential statistics. And under the descriptive statistics, we tend to summarize data. So depending on the domain or the field you find yourself, you could have different types of data visualizations. So with the descriptive, we visualize data, we summarize data with percentages, we find means, medians, and so on and so forth. So that is basically what descriptive statistics is all about. But one thing we have to notice that with descriptive statistics, we cannot make inference at that stage. So for instance, if you look at the figure here, the top left one, you realize that, I mean, we could see a decline in terms of these S protein levels, for instance. However, we cannot say that there's a significant decrease in terms of the S protein across the population until we do an official or until we do a statistical test. We cannot make any conclusion. We can only say it appears that is the pattern we are seeing. So this brings us to why we now need to think about something called inferential statistics. So under inferential statistics, we are basically now trying to use either statistical tests or statistical models to now make inference. So it means that it is only after we employ a statistical test or a statistical model that you can now make vivid and meaningful deductions and conclusions about patterns or about research questions we want to identify and solve. So if you look, so it's imperative that if you visualize and there's a way of adding the results of a statistical test to it, it makes it more meaningful as opposed to just having a graph without any statistical test on there. So this is just trying to say that every statistical analysis, even in most research, we often summarize the data in the best possible way, visualize the data, and at the end of the day, we try to now answer some questions. And that stage, we need inferential statistics to achieve that. So it is important. There are some few terminologies that we need to have, try to familiarize ourselves with. Some of them might be known to you already. So often when you talk about parameter, so you realize that parameter is any measure we compute from a population. So when we say a population, it means that the total number of entities or group of interest. So a parameter is any measure we compute from a population. So the population could be any target population of interest. However, when you consider a subset of the population and decides to compute that same metric or that same measure, then that measure is now a statistic. And one thing we have to notice is that there are different types of variables, and it's very important to know the type of variable. And then because the type of variable could also sometimes completely inform the kind of statistical methods we could use. So how many types of variables do we have? So broadly, we have quantitative variables or quantitative data and qualitative data. Another name for quantitative data or variables are numerical data. And qualitative, you could also call it categorical. So it means any variable or data that is categorical in nature is known as qualitative data. But one thing you have to notice that if you have, let's say, a variable like so for qualitative data, they do not naturally take up numbers. But quantitative data, they mostly naturally take up numbers. So if I ask you of your age, you are going to give me a number. So that makes age numerical or quantitative. But one thing you have to notice that if you have, let's say, a variable like sex, which is, let's say, male and female, that is a categorical variable, so it's qualitative. But assigning a dummy code that is 0 and 1, let's say whenever you see a female, you say 0, a male, you assign 1, does not make it numerical. So this is to say that assigning numbers or dummy codes to qualitative data does not transform it into a numerical data. This is something we have to really bear in mind. And because most of the times we confuse that. Now, we have also scales of measurement. So basically, we have 4 scales of measurement. They are imperative, something you need to know. So for the qualitative data, we have 2 types. So we have nominal. It could be either on the nominal scale or the ordinal scale. So for the nominal scale is just instances where characteristics are just by names, and then there's no ordering. So when I say gender, male, female, you don't say male is better or greater than a female. So there's no ordering there. For ordinal scale, that is when a nominal variable or a nominal scale can now be ordered. So if you think about, let's say, educational level, basic, primary, tertiary, we can see some ordering in there. So that makes it ordinal. And keep in mind that if you are doing an analysis and then your dependent outcome or the outcome variable is nominal or ordinal, it can inform different choices of methods you want to use. And so for the numerical one, it's either on the interval scale or the ratio scale. I wouldn't go much into that, but the clear distinction is that for the interval scale, the absolute 0 does not make any meaning. So for instance, does it make sense if we say someone's height is 0? It's not possible that anyone's height could be 0. So in that case, it means height of humans is on a continuous scale. If you think about the number of, let's say, road accidents or the number of genes or the number of cells, if the number of cells or the number of something is 0, that 0 makes a meaning, isn't it? So in that case, it makes such variables to be on the ratio scale. So it means for ratio scale, one clear distinction is that they are already on an interval scale where the 0 of such a variable makes meaning. So that is basically the clear distinction. So now, another most important thing, so after understanding the types of variables we have, whether it's numerical or categorical, because as I said, it's going to completely inform the choice of methods we can use, it is also imperative to know that the structure of the data or the structure of data we use is also imperative. I know, depending on the domain or the field you find yourself, you would have different types of data. Some are into genomics, some are into proteomics, and so on and so forth. But so here I'm just trying to use a simple scenario to just describe. So broadly, we have 2 or 3 types of data structure. Of course, we could have several types of data structure, but for research purposes, we can just say that we have broadly 2. So we have cross-sectional data. So cross-sectional data is any data where you collect several information or several variables such that for each row of your data correspond to a particular subject. So often we collect such data within a certain time frame. If you think about it, every data could be indexed by time because at any point in time, you are collecting data at a certain space in time. However, if you are saying that within a certain year, you are collecting information about, let's say, the wage, other information, or it could be some health variables. In that case, if for each row of the data correspond to a particular patient, then such a data is called cross-sectional. However, if you decide to now collect that information, the same set of information in 2013, in 2014, or basically trying to collect the same information over time, then it makes it something called a longitudinal data. So it means for a longitudinal data, an individual could have multiple rows, whereas, so here we have 2 rows for this particular patient or individual. But for a cross-sectional, every single row corresponds to a particular type of individual, not necessarily individual, it could be a subject. So let's say if you have in genomics, or let's say in RNA sequencing, where you have a data concerning, let's say, genes, and then each row corresponds to a particular distinct gene, such a data could be seen as a cross-sectional. Okay, but there's something called pool cross-sectional. So we realize that for the cross-sectional, we collect the data within a certain time frame. So yeah, a specific time frame. But situations where we decide to collect some set of information for a particular individual, a particular set of individual within, let's say, in 2000, and then within the year 2013, we decide to collect the same set of information across among different set of individuals. So you realize that still each row corresponds to a particular individual, just that the time frame within which the data was collected were different for some patients or for some individuals. And that is what we mean by pooled cross-sectional. So still a pooled cross-sectional data is still cross-sectional because each row corresponds to a particular individual. And then a time series is basically when you collect data over time. So in essence, if you combine a cross-sectional data which has been collected over time, then we have a longitudinal data. And it is important to know that if a data is cross-sectional, the class of statistical method and test you can adopt is completely different from when it's longitudinal. So that is why it's important to know. So now we are just going to touch a bit on some univariate methods and the multivariate methods. But here there is a disclaimer I want to make. When we talk about univariate analysis, often one thing we have to notice that there is a particular outcome variable, a particular dependent variable, and it's just one. So situations where you are trying to do an analysis where the goal is to look at maybe the mean difference or the effect of some variables on a single outcome, or you are trying to look at how a certain outcome variable varies between groups. In that case, you are trying to do univariate analysis. So anytime you hear the word univariate, it means single outcome. When you hear multivariate, it means multiple outcome. By outcome, I mean multiple dependent variable. So there are so many statistical tests, but before then, what is the difference between a statistical test and a statistical model? Sometimes people confuse the two. Now you realize that for statistical tests, the goal is to test a certain research hypothesis. So it means for statistical tests, we are trying to ascertain the validity of a certain hypothesis, whether it is true or not. I mean, by hypothesis, I mean an assertion or a statement, which could be true or not. And the goal is to ascertain its validity. But so it means that under statistical tests, we don't estimate parameters. So it means we don't estimate. We are rather testing whether the population parameter is true or not, depending on the kind of hypothesis you are trying to do. But for statistical models, there are always parameters that we need to estimate. So it means that when you talk about regression model, it means there are parameters, and that makes it a model, and those parameters needs to be estimated. But when you talk about a chi-square test, we are not estimating any parameter. We are basically testing some hypothesis. So that is just the clear distinction between statistical test and then statistical models. So basically we can combine statistical tests and models as statistical methods. So when you say statistical methods, then in this case it's actually coupling statistical tests and statistical models. But keep in mind that they work hand in hand, such that when you fit a statistical model, okay, or when you have a statistical model which has parameters, after estimating those parameters, we have to check whether those parameters are significant or not, and then we rely on statistical tests. So it means statistical models cannot act alone. It always requires statistical tests to make it complete to some extent. So here we are just going to summarize a few of the statistical tests we can do. So of course, I'm sure we all know about situations where you are trying to compare means, and then you have just a single population, and you are trying to compare whether the mean is going to be equal to some hypothesized mean or not. So that is like a one sample t-test. And here, its non-parametric version is either the Wilcoxon sign test or the sign rank test, which is also Wilcoxon sign rank test. So here I've just summarized for each test its non-parametric version. But here there's the take-home message is that for parametric test or parametric models, okay, one thing is that there are very strong and specific assumptions that needs to be met. So it means anytime you are trying to use a parametric test, don't use it without testing its parametric assumptions, okay? Because at the end of the day, once the assumptions are violated, parametric test or models can be completely useless if you think about it in terms of its accuracy. Nonetheless, non-parametric tests are quite robust to the departures of these parametric assumptions, and such that whilst in terms of the mean test, for instance, let's say you are trying to test means, but one thing we notice that when the data is not normal, meaning that when the data contains outliers or when the data is skewed to some extent, what happens is that you realize that mean is something that can easily be affected by outliers, right? So for instance, if I have, if I'm comparing, let's say, two classes, in terms of a score of two classes, and then we have maybe 3 students in each class, and then we have one class getting a mark of, let's say, 10, 20, 30, and then we have another class where students are getting, let's say, 10, 30, 20 is not the best example. Maybe let's say 50, 40, and then one gets 90. And then another class, people are getting 70, 80, and let's say 60. If you compare the mean, you realize that because of that outlier 90 in that other class, you are more likely to say that the class where some student had 60 and 50, just that one had 90 was doing better than the other. Meanwhile, it's just because of an outlier. So in essence, because means are affected by outliers and means are affected by skewness in some essence, that is why it is always imperative to ascertain whether the data is normally distributed or not. And if it is not normally distributed, you don't need to worry about testing means. You can test medians. As a matter of fact, when the data is normally distributed, the mean and the median are the same. So why then do we? So it means medians are more robust to these departures. So if you think about it, if I have 20, 30, 40, if I should find a median, it's still the middle. If I replace the last number by a very large number, the median will still be the middle number. So basically, medians are less affected or often not affected by outliers, if you think about it. So the paired t-test is situations where you have dependent samples, meaning that if you consider a certain set of individuals, you recorded some information about them at a certain time point and decided to record the same set of the same information at another time point, then we have a dependent sample in this case. And in that case, if you want to compare the means of that variable between the two groups, then we now have something we call a paired t-test. And its non-parametric version is still the same with constant sign test or the constant rank sum test. But situations where we now have two independent groups, let's say male, female, and you're trying to check whether the average outcome is significantly different between these two groups or not, then in that case, an independent t-test becomes the best choice. That is, if the data is normally distributed. But if the data is not normally distributed, then the Mann-Whitney test or the rank sum test becomes the alternative. And the same applies for ANOVA. ANOVA extends the independent t-test, where now you are trying to test a certain continuous outcome just to see that whether its mean is significantly different across 3 or more groups or 3 or more independent groups. And if the parametric assumption of normality is not achieved, then in this case, the Kruskal-Wallis test becomes an alternative. But what about repeated measures ANOVA? So there are instances where for the same outcome variable, you recorded that information at multiple time points. So if you think about it, it's just like a paired T, it's just like the paired case, but just like the paired case, you recorded the data at 2 time points, or 2 instances. But situations where you recorded at multiple time points, okay, in that case, and then you're trying to compare that's the mean of that outcome, which was recorded as a repeated measures across 3 or more groups, then we have a repeated measures ANOVA. And its non-parametric one is called Friedman test. So I wouldn't go into all the details. So here is just to summarize some of the parametric tests and their non-parametric version. But here there's something I want to point out under the correlation test. I'm sure most of us have heard of Pearson correlation and the Spearman rank correlation. So basically, the Pearson correlation tends to look at the linear relationship between two variables, specifically two continuous outcomes. But and most of the times we say that when the two variables are categorical, we use a chi-square test, isn't it? But one thing we have to notice that there are also instances where you want to test the relationship between a continuous outcome and a dichotomized variable, meaning that you want to check if there's any relationship between, let's say, which continuous variable can I think of? It could be a gene expression. You want to check if there's any significant, there's any correlation in terms of the gene expression between male and female. So one thing you have to notice that there are two types of correlation that achieve that. So we have the point biserial correlation and we have the biserial correlation. So situations where the two categorical, that categorical variable which two levels has a natural, that is, has a discrete or true dichotomy. By discrete or true dichotomy, I mean an individual could either be infected or not. So either you are dead or alive. So when you have such a variable and you want to check how such a categorical variable with such true dichotomy is correlated with a certain continuous outcome, one could now use what you call the point biserial correlation to achieve that. And then the biserial correlation is when the dichotomy is not, it's just continuous, or it's not true, or it's false. In essence, situations where there's continuum between those two variables. So for instance, instances where you're not too sure how to split a continuous variable into a binary outcome or a binary variable, and then you try to use median split, or you try to use another metric, which is not a true representation of or as a splitting metric, then in that case we have a continuous dichotomy. And in that case, if you want to check the relationship of such a variable with a continuous outcome, you then go for a biserial correlation. Now, partial correlation is something you often see when you do regression. But so one thing about partial correlation is trying to look at, so you realize that for the Pearson and the other ones I've talked about is looking at the bivariate relationship between two outcomes in the absence of other variables. So partial correlation is when you are trying to look at the correlation between two continuous outcomes or two variables, whilst controlling for the presence of other variables. Then such a correlation is a partial correlation. So that is one thing I wanted us to note. Now, in terms of when you have two categorical variables, whether the levels are two or more, and you want to test the relationship, the association, then the chi-square test of independence is what we often use, and its non-parametric regression is what it's the Fisher's exact test. And then, so we have chi-square test of homogeneity. Sometimes people confuse that with the independence. The independence is looking at association, whether two categorical variables are associated or not. Whereas the homogeneity is situations where you have a certain categorical variable and you want to see if the levels of another categorical variable is significantly the same or not across the other variable. So in terms of chi-square test of homogeneity, you could see it as an extension of the two sample proportion test, where you are now trying to test for multiple proportions whether there's significant difference or not. And again, its non-parametric test is what has been specified here. But there's one thing that most people should know. Is called the ANCOVA. So you realize that analysis of variance is what we call ANOVA. ANCOVA is analysis of covariance. And it is one method that is very useful in many disciplines, but most people are not aware of, or they hardly use it. So analysis, you realize that for ANOVA, we are looking at whether there's a significant difference in the mean of a certain continuous outcome across three or more groups without necessarily controlling for other potential confounders or other extraneous variables in that case. So for ANCOVA, what it does is that it employs both ANOVA and then it employs a regression technique. So it means that any regression model you've seen before, the situations where you are trying to look at if there's any significant difference in some continuous outcome across three or more groups, whilst controlling for other potential controlling variable or confounders, then in that case, we have an ANCOVA. And another instance is, there are instances where instead of using a repeated measure as ANOVA, one could think about ANCOVA in this case. So instances where, let's say, you've you've recorded a certain outcome at a certain time point. That is at baseline. And then you also have information recorded at a certain future time point. And then you wanted to check whether the data at that future time point varies across some groups whilst controlling for other potential variables. What we need to do is also to adjust for the baseline score. So it means for ANCOVA, we expect to see another outcome that co-varies with the another predictor that co-varies with the outcome variable. And one easy choice is a baseline score, which at this case you are trying to adjust for that baseline score in your regression. So it means if you think about any regression model where you have a certain outcome at a certain future time, and then you have the baseline score as a predictor in the model whilst controlling for other variables, and you have some specific predictors. In that case, we have a classical ANCOVA. And in an ANCOVA case, you can also adjust for interaction effect and so on. So it means ANCOVA extends the classical ANOVA. And everything I've said here, and then in terms of the ANCOVA, we have its rank, we have its non-parametric versions, which I've highlighted here, preferably the rank-based ANCOVA. And all these I've mentioned is assuming that we have only one dependent variable. But what if we have multiple dependent variables? Now, that introduces us to something called multivariate statistical test. But one thing I want us to touch on is multiple testing. So you realize that there are instances where you have set of hypotheses, like you are trying to test a certain outcome simultaneously across some groups, whether there's significant difference or so and so on. So we tend to do multiple testing. And when you do multiple testing, what happens is that if you don't properly adjust for its limitation, what happens is that you lose statistical power, and also you introduce something you call lot of false positives, or you introduce a lot of false discovery rate and so on. So what people do is that if you have, so this brings us to what we call p-values, and another thing called k-values. I'm not sure how many have heard k-values, but k-values are also is basically an adjusted p-value, and which has been adjusted for multiple testing. So when you talk about p-value, you realize that p-value is just a measure of evidence. Okay, it's just a measure of evidence in an hypothesis testing. Okay, so that is basically what p-value is trying to do. So it means that situations and p-values we often affiliated to a single hypothesis. So situations where you are now testing for multiple hypotheses in the same scenario simultaneously, then what others do in terms of using a certain correction called Bonferroni correction is that you compare the p-value to the significance level. And we know that if your p-value is less than your alpha level or your significance level, we tend to reject the null hypothesis. But if it is greater than the alpha level, we fail to reject the null hypothesis. But situations where we have multiple testing, Bonferroni correction suggests that divide the alpha by the number of multiple hypotheses you are trying to test, and that brings us to that, and that is basically a way of trying to adjust for or control the power and also the type 1 error rate. However, k-values basically what it does is that it's a measure of the proportion of false positive or false discovery rate. So in essence, it is much more improved version of just trying to use a p-value with a Bonferroni correction in terms of the alpha. But in terms of the k-values, what it does is that it modifies the p-value itself to account for multiple testing, and then you can now compare that with a certain threshold, a false discovery threshold, which often is also set as an alpha level. So it's just something I wanted to touch on, that though multivariate analysis is important, I'm just trying to say that if you are still trying to use univariate methods to still do things that multivariate should have achieved, then you have to properly account for such multiple instances as well. So that introduces us to k-values and p-values, which you can read further on that. So now for multivariate methods, there are instances where, let's start with MANOVA. So MANOVA is multivariate ANOVA. So you realize that for ANOVA, we're just looking at one outcome. But situations where you have multiple outcomes and you're trying to check whether that vector of outcomes significantly differ across some groups or not. In that case, then we say that instead of doing multiple ANOVAs, which loses power and introduces a lot of false positives for among significant tests, then you should rather go for a multivariate analysis, so a MANOVA. So it means that at any point where a multivariate statistical test is applicable, always do that first. If you think about it, if you are doing an ANOVA test, the only instance ANOVA test will conclude a significant difference is if between any pair of your categorical variables. So for ANOVA, we are comparing 3 or more groups. So if you take any pair and there's at least a significant difference between any pair, then the overall ANOVA test will conclude on a significant difference. So it means the moment you do a univariate ANOVA and it says that there's no difference, then it means that there's no pairwise difference between any pair of the categorical variable. So that is employing that same idea. Before a multivariate ANOVA will conclude on a difference, that there's a significant difference, then it means that at least one of the outcome was significantly different across the groups. So if you think about it, if MANOVA concludes that there's no difference, then there's no need of doing univariate ANOVA. So that is why instead of wasting so much time to do univariate ANOVA and then do multiple comparison, in instances where you are trying to test multiple outcomes, how they differ across a certain categorical variables with 3 or more levels, then go for a MANOVA, so that in that case, if the test concludes there's no significant difference, then it means that none of the univariate outcomes or none of the univariate ANOVA tests would have concluded on significance. So it saves you so much time, and at the end of the day, it preserves power as well. And then you don't need to worry about correcting for multiple testing and so on. So that's the advantage. But keep in mind that it's expected that the data should be multivariate normal. Keep in mind, every parametric test has parametric assumptions, which has to be achieved. But of course, with MANOVA, even if the data is not multivariate normal, it has different test statistics. It has something called Wilks lambda. You don't need to understand, and it has a Pillai trace statistic. The Pillai trace statistic is robust to departures from non-normality. So I'm just trying to say that for MANOVA, it still has, though it's parametric, there are some test statistics that you can still report in the presence of departure from multivariate normality. However, again, the multivariate Kruskal-Wallis test is also there to test for the same thing in the presence of non-normal multivariate outcomes. So just borrowing the idea of ANCOVA, we can also have MANCOVA. So MANCOVA in this case is multivariate ANCOVA, where in this case you have multiple outcome variable that you are trying to check how they differ across some groups whilst adjusting for the effect of other controlling or confounding variables. So that is what ANCOVA does. So, and I mean, with time we'll hear about multivariate regression analysis. So it means that we are going to talk about regression very soon, and what we are going to talk about, we'll start with univariate and then we'll talk about multivariate regression. But here, let me just give a disclaimer so that I don't repeat myself when I get there. So when you talk about a simple regression means that there's only one. So keep in mind, regression is when you have a certain outcome variable and a set of predictors. Okay, so predictors are also known as explanatory variables, or also known as independent variables. If we have only one predictor or one independent variable and a single outcome, we have what we call a simple regression. The moment we have a single outcome but several predictors, then we have a multiple regression. So if you talk about multiple, so people confuse multiple regression for multivariate regression. The moment you hear multivariate, it means multiple dependent variables or several dependent variables. So it means that we could have a multivariate simple regression. It means it's a regression model with several outcome variables or several dependent variables, but only a single predictor. So in that case, that is a multivariate simple regression. So we could actually have a multivariate multiple regression where we have several outcome variable and multiple independent variables. So anytime you hear multiple regression, it's rather the predictors you should think about, whether the predictors are more than one or not. So that is where multiple comes in. But multivariate is always affiliated to the outcome variable. Though in survival analysis, sometimes we assume that Kaplan-Meier method, for instance, is more like a univariate case, and in the process of trying to now adjust for other controlling variables or confounding variables or adjust for other predictors using cost proportional, then becomes like a multivariate case. But if you still think about it, it's not really multivariate. Probably we could call it multivariable survival analysis if you think about it in that case. But multivariate is often attributed to multiple outcome variables. And there are so many other multivariate methods we can think of. I will not go into the details of all because of time, but principal component analysis is often used a lot. But keep in mind that is a variable, it's a data reduction technique. It is not a variable selection technique. People confuse the two. So variable selection, we'll talk about it soon. And then, but keep in mind for principal component analysis, it tries to reduce the dimension of a data where the data are all numerical in nature. But situations where we have strictly categorical data that we want to still reduce the dimension and identify even potential latent effects or control for the effect of other latent variables, then factor analysis becomes imperative. So it means factor analysis is quite similar to principal component analysis in that the difference is that one is strictly on numerical variables, but one is strictly on categorical variables. And linear discriminant analysis is when you want to now look at how a certain variable discriminates the effect of some outcomes and on some predictors. So I won't go much into all these details, but these are just methods that you could read into. And keep in mind, your knowledge of these statistical methods can inform even how you want to design your study. It could even inform the best way you could answer some research questions. But again, at any point in time where multivariate method becomes an option, always go for that, because in that case, you don't have any issues about effect of multiple testing. Somehow it improves power. And also think about degrees of freedom. I'm sure you've heard of degrees of freedom. Now, if you think about degrees of freedom in this way, if you have a model, if you have a statistical model, it's not always that you are interested in estimating all the parameters. So there are instances you are just interested in estimating a subset of the parameters. So if you look at the difference between the total parameters that the model can possibly have, and you take away the parameters that you want to estimate from that pool of parameters, that difference is like the degrees of freedom. So the degree of freedom is like those other parameters that is virtually not going to be touched. Right. So if you do lots of univariate methods, it means that you tend to overestimate parameters, whereas in the multivariate case, you control for not having to estimate a lot of these parameters. So somehow it also conserves your degrees of freedom as well, depending on what you are trying to do.